My name is Yehuda Carmeli. I'm coordinating together with Vincent Javier the session on global spread of carbapenemases. And the first speaker will be Tim Walsh, the man who brought us NDMs. Without him, we would not have it.
Thank you, Yehuda. I didn't bring it personally. I just want you to reassure you. It was just some science. I'm always reminded of a quote by Shakespeare. If you're talking in an auditorium and it's at least half empty, he once said, "We happy few," which was a quote from Henry V. And it doesn't really matter on the quantity, it's the quality that matters. So I'm going to be talking on kind of the global spread of carbapenemases. And this, I've got about 30 minutes, really, or 25 minutes to talk about the epidemiology, about human dynamics in the spread of it, a little bit about the molecular biology, because it's important, and also about methods of detection. So you'll forgive me if I'm parsimonious in many respects to this, and I think it's just important that we kind of, I lay the basis of the foundation for the next speakers. So let's start with a quote from Aristotle. "The only thing I'm certain about is that I know nothing." And the more you learn in science, the more you realize you know absolutely nothing. So as a PhD student, you think you know everything, and then one day before your retirement, you realize you know absolutely nothing. And a very good friend of mine added this to the quote, "And even then I'm not sure." And if Fernando Bequeiro is not sure that he knows nothing, then I definitely am unsure I know nothing. And I think this kind of reflects what we see with the increase in resistance in gram-negative bacteria. It's kind of got ahead of the game, and we've been left behind in the race in trying to catch up and understand how resistance occurs and how it occurs at such a very rapid level. This is a slide which I'm sure you've all seen before, and basically some of the problems that we had with the third generation cephalosporins, whereby we then had CTXM and ESBLs, then some countries around the world were then using lots of carbapenems here, and then we got the carbapenemases. And at the moment we're probably up here now with almost pan-resistant Enterobacteriaceae in some countries. And I'll talk about the KPC issues and NDM1 in a moment. So what is a beta-lactamase? I appreciate that you all come from various backgrounds, and some of you are from infection control, some of you may be nurses, some of you may be clinicians and even don't know what a beta-lactamase is. So basically, a beta-lactamase is simply an enzyme that destroys a beta-lactam molecule. So this beta-lactam ring here is part of your overall beta-lactam molecule. And this could be a penicillin, a carbapenem, or a cephalosporin. They always attack here, in this little bit here, in the sigma bond to the oxygen. And it's an anion attack that takes place, and it involves water, and the mechanism is what we call hydrolysis. When that actually happens, you get water molecules added onto the nitrogen and the oxygen. You then have the splitting of the beta-lactam ring, and at that point, the molecule becomes inactive. So there's 2 points to make here. First of all is that your hydrolyzed beta-lactam is no longer active, but importantly is that the enzyme produced by the bacteria is then free to go and then hydrolyze other molecules, beta-lactam molecules. And so the bacteria actually doesn't have to produce an awful lot. All it's got to do is to produce enough to make sure that it can reduce the active beta-lactams and keep it down to a level such that it will not be killed. So if that is a beta-lactamase, what is a carbapenemase? And a carbapenemase, by definition, is an enzyme that hydrolyzes carbapenems, ertapenem, imipenem, meropenem. So how much does it have to hydrolyze for it to be called a carbapenemase? And incredibly, nobody knows, or we haven't actually decided. It's a bit like the European Union voting on the size and shape of bananas. There's various opinions on that. And so we came up with a definition a while ago, rightly or wrongly, that in order for an enzyme to be called a carbapenemase, it must actually have the capacity to hydrolyze carbapenems at this particular rate. And so what we've tried to do is to try and put something sensible behind some of the definitions. So are carbapenemases ESBLs? ESBLs are classically described as enzymes that hydrolyze oxyaminocephalosporins and are sensitive to clavulanic acid. That is the classic definition of an ESBL. But actually that precludes oxyacylinases, it precludes class C, mobile class C enzymes, and it also precludes metallo-beta-lactamases. Now let me give you a definition. Supposing that half of you in the room owned a Skoda, and the other half in the room owned a Mercedes. And the ones in the room decided, well, I'm not satisfied with my Mercedes, and therefore I'm going to make it into what they call a Mercedes McLaren. I'm going to give it a bigger engine, a better suspension, whatever. And then you turn around to the people in the room who have the Skoda, and you say, look, I have a Mercedes McLaren. And they say to you, no, you don't. You have a Skoda. And it's a bit like the same definition with ESBLs. So you actually have enzymes that can hydrolyze oxyamino acids that are called ESBLs. But when they have the capacity to hydrolyze other beta-lactams like carbapenems, all of a sudden they are not ESBLs. And there's a real problem with the definition. The problem comes from the fact that 20 years ago this definition took place. It was actually 21 years ago, when there were only a handful of ESBLs, 3-TEM, 5-SHV. Unfortunately now there are several hundred of each of these variants, and therefore unfortunately the definition hasn't moved on, so it's very confusing. And here I just illustrated the fact that these here are ESBLs, and guess 2, guess 4, guess 5, and guess 6 are carbapenemases, and yet still guess 5 is referred to in the literature as an ESBL. So if you're confused, so am I, don't worry. So we go back to that definition of Aristotle, okay? The only thing we know is that we know nothing. So I'm really just going to concentrate on 3 groups of enzymes, because we could be here all night, or all day and night, KPC2/3, OXA48 and its variants, VIM2 and NDM. I'm not going to talk much about NDM because it's being talked about a lot. So these are the metallo-beta-lactamases. And this isn't meant to be absolutely inclusive of everything that's been reported. I really just want you to get a feel for where we are in terms of the world map of MBLs. And I'd just like to draw your attention to 2 in particular. First of all is VIM. Now, VIM2 has occurred in Pseudomonas aeruginosa. Very rarely does it occur in Enterobacteriaceae. Sometimes occurs in Acinetobacter baumannii. But if you have a look at the blue dots spread around the world, the majority of those make up VIM2 in Pseudomonas aeruginosa. And I've got some data on that in a minute from Russia. The next thing to note is the light blue triangles, which are NDM. Now, 3 or 4, well, actually 2 years ago, NDM didn't exist. It was off our radar, off our scientific radar, but you can see now that actually it's done a very good job in spreading very widely, and that's pretty much to human travel, exclusively indeed to human travel. So this is just some data, quite old data now, and we're doing a lot of work with the reference laboratory up in Smolensk in Russia. And this is some data from Mikhail Edelstein, who runs the reference laboratory in Smolensk. So this is the situation in Russia in 2002-2004, where we had 4.6% positivity MBLs. This is the situation in 2006-2007, where that positivity now has gone up to 20%. We now look at 2007 and 2009, and we see a massive spread of Pseudomonas aeruginosa carrying VIM2. It's carried not on a plasmid, but on a chromosome, and it looks like you have a colonial spread, if you like, of prominent clones throughout Russia. We're not too sure how this happens, but we suspect that given the sort of geographics, if you like, of Russia, that it's probably actually spread by wildlife, birds, and in particularly geese. So this is kind of very worrying at the moment. And again, this is old data, 2006, 2007. We're still trying to characterize the strains that are coming to us, and we hope to publish very shortly. But I'd just like to draw your attention to Novosibirsk. Have a look at this. 100% of their Pseudomonas aeruginosa contain metallo-beta-lactamases. And that was 5 years ago. And the data with these other hospitals or other areas has increased. Okay, so it's just not NDM. We shouldn't be NDM myopic. But in fact, Vintu in Pseudomonas aeruginosa in some countries poses as big a problem. So this is NDM, and this slide you may have seen before. And I just really want to draw your attention to sort of the focal point in Europe, at the moment at least. Where we have the red stars is almost directly related to Southern Asia, and in particular India, either through travel, normal travel, or indeed through medical tourism. Kenya certainly is related to India, and I'm completely convinced about that. China seems to only have Pseudomonas aeruginosa, Acinetobacter baumannii at the moment, containing NDM, and it most certainly started in Acinetobacter baumannii, but probably didn't start in China. We don't actually know where it started. But the interesting thing is the Balkan region, the countries here, there's no epidemiological linkage to Southern Asia, at least direct linkage, at least to India. But we actually think that there's a good train of thought, and we're working with several journalists at the moment to actually link these cases here with the illegal trade of organ transplants coming from Pakistan. Again, this is not meant to be completely inclusive, and you can just see here that KPC 2 and 3 on Carbapenemases, which I think Yehuda will talk about shortly. But it's interesting to note, where did it start? Did it start in America, or did it start in Israel? The Israelis might blame Americans, and the Americans would blame the Israelis, and we'll seek further clarification of that in about 20 minutes or so. But nonetheless, it's actually been quite successful. It was slow to take off. The first case was reported in 1996. KPC2 was reported in 1997. And so it actually, there was a lag phase in the way in which it was able to disseminate around the world. And the reason for that is the fact it didn't find the right plasmid backbone. The reason why NDM has been so successful is because very quickly it found a variety of plasmid backbones that would then allow it to disseminate into lots of gram-negative bacteria. KPC has now found an AC plasmid. The marriage took place about 2 years ago and was reported, I think, the beginning of this year. And so therefore we can look forward to KPC perhaps turning up in Vibrio cholera, rather like NDM1 has. This is just some data from the Swedish group who we do some collaborative work with, and this is kind of just probing. And here you can see the plasmids very clearly, and what we've done is to probe it. This is the southern hybridization map, and you can see that some strains have more than one KPC2 gene, and in fact, some actually have KPC3 in Sweden as well. Often as not, with these bacteria, you'll get multiple plasmids and multiple copies of the same gene, and that kind of confers an increase, if you like, resistance. Oxotype carbapenemases, these kind of, again, had a lag phase. This group up here, whoops, I'll go back one. This group up here, the blue and the red, are pretty much exclusive to Acinetobacter species, and indeed Acinetobacter baumannii in particular. But these ones here, the green and the light blue, are exclusive, well, pseudo-exclusive, if I can use such a term, to Enterobacteriaceae. And there seems to be sort of an awakening, if you like, of OXO48, particularly in Enterobacteriaceae. OXO181 is actually an Indian variety of OXO48 with just a few amino acid changes. And this is actually widespread pretty much all over India. There is another variety that has recently been reported about a few months ago in Southern America, particularly in Brazil, which is OXO163. And again, that's a very close variant of OXA48. This kind of just shows you and I was offered this slide, and I acknowledge Patrice Norman in this. This kind of shows you the antibiogram of OXA48 containing isolates. And you can see here that actually they don't confer very high level resistance. And that's the problem with both metallo-beta-lactamases and serine carbapenemases, is that when they're sometimes in Enterobacteriaceae, the MICs are quite low and actually either straddle the breakpoint or actually are below the breakpoint. So in terms of detecting them and trying to maybe isolate the patient and have good infection control measures, it's a very difficult thing to do. This is kind of where we are now with OXA48, looking at a fairly detailed map. We have them in Wales, they're in the UK, and we have some recent imports in France from both Morocco and Turkey. And so rather like NDM, you can use it as a molecular marker for patient or travel, just through tourism, for instance, from fairly high density areas such as this into, at the moment, areas where the prevalence is quite low. So these organisms contain plasmids, and that is the single reason why they're so successful. Bacteria like to have sex, and so we can do this in the laboratory. We turn the lights down, we play some Mozart, and 20 minutes later, it's all happened. And I'll leave it there. So it's very easy to do, and in fact, you don't need laboratory recipient strains. You can do this with clinical isolates as well to have a look at the promiscuity, if you like, of strains to receive plasmids that have lots of antibiotic-resistant genes. But as I said before, sometimes some plasmids are more successful than others, particularly going into non-Enterobacteriaceae species. And again, INC-AC plasmids are particularly good at this. So this just shows you, this is basically the parent and the transconjugant and the parent and the transconjugant. So what I mean by that is the parent is the donor and the transconjugant is the recipient or the female, if you like. And you can see here that actually the plasmids change size. When it's gone from one bug to another, it's got smaller. And that shows you that these systems are very fluid and very plastic. And they're not stable. They change all the time. I don't know if any of you have seen an aurora, like the aurora borealis up in Norway. It moves. And that's just what DNA does all the time throughout bacterial populations. It moves. It never stays still. And this is really interesting, again, where we have our donor and our recipient. In this case, we have an increase in DNA, in size, in DNA. We have accumulation of DNA, either from another plasmid or from the chromosome. And this is very rarely reported, and our understanding of plasmid biology and how this occurs and why it occurs is still very embryonic. I'm just showing you this because some strains have more than one plasmid in it, type of plasmid, carrying the same gene. And some will actually have it on the chromosome as well. And you can see here that there is a plasmid down here where the copy number, in other words, the amount of gene, is about 20-fold. And we're actually sequencing that at the moment. So remarkable fluid systems, these small plasmids and bacteria. And if only we could catch up with them in terms of the evolution, if you like, of our DNA. So this is kind of one of the main mechanisms of how DNA spreads. And these are gene cassettes, and this is an integron, and this is the first, one of the first European VIM2s, and this is, again, one that was very close to it, the Polish VIM2. They're actually quite related. You can see structurally they're related. And what happens is we have importation of a gene and then excision of a gene. And again, this is very fluid system. So if you treat the patient with gentamicin, they may pick up an AAC-A7 gene and it be inserted into this particular structure, which we call a class 1 integron. Now, 15 years ago, class 1 integrons were not very common. But I would bet anybody in this room that if you went back and screened your Enterobacteriaceae for class 1 integrons, about 70%, 80% of your E. coli would carry a class 1 integron now. And so the prevalence of this has gone up remarkably. Sometimes these class 1 integrons are embedded in transposons. And transposons are useful because they are what we call jumping genes. They're able to take DNA from one area and move it to another. So for instance, this is the integron here, and this is the transposon here. Now, sometimes that transposon can go over there or over there, but actually the integron itself is not able to move. So in order for it to be able to move, it needs a transposon. And again, the prevalence of transposons in clinical isolates has gone up tremendously in the last couple of years. And here I've just shown you a metallo-beta-lactamase, or 2 carbapenemases, intrinsically associated with integrons and transposons. There are other elements, and this is, I think, the second last slide that I'm going to show you on genes, so bear with me. And this is really interesting because there was a transposon called TN402, and we forgot about it. Everybody ignored it. Well, interestingly, about 3 or 4 years ago, in fact, it was an Italian group that first identified it, although they probably actually didn't know the significance of it. You have actually importation of a class 1 integron into this ancestral, very ancient now transposon 402. And now that we actually know it's present, we've gone hunting for it around the world, and it's in Russia, it's in China, it's in India, it's in South America, and so we actually didn't know about it. It was off the radar. And that's one of the things that's interesting about molecular biology. It always teaches you that you know absolutely nothing. And so we have, other systems that we can now recognize as actually causing clinical antibiotic resistance. KPC3 also has such a system as well. In this case, they called it TN4401, and here we have KPC3. And KPC3, and indeed sometimes KPC2, are intrinsically linked to TN4401. That's able to mobilize this from plasmid to chromosome, and then back to plasmid, and then via conjugation or bacterial sex, transfer the plasmid. These structures are very complicated, and this just shows you this slide. In fact, the next 2 slides are simply to illustrate how complex it has all become. So here we have a group of carbapenemases. In this case, they're all metallo-beta-lactamases, and this is some beautiful work done by the group down in Madrid. And these basically gene cassettes sit in these integrons here. These are the bacteria, and these are basically the transposons that carry them. So it's really like a genetic puzzle, but it's like a genetic puzzle where the bits of the puzzle can actually almost fit anywhere. Again, this just shows you the transposon, and you have a look at those class 1 integrons that are actually carried by the transposons. These are the metallo-beta-lactamases here, and you can see just how varied the gene cassettes are. One of the important things to point out, and I made the comment earlier on, but I want to reiterate it, is a lot of the time that metallo-beta-lactamases in Enterobacteriaceae do not confer resistance, and neither does OXA48, and not always does KPC. And so here we can see the MICs, for instance here, are actually below the breakpoint, even the new breakpoint. And so it's very difficult sometimes to detect them. Do they mediate clinical resistance? And this is a beautiful study done by the group in Athens. It's actually one of the seminal papers, and I remember reviewing it many years ago when I was a reviewer for ISCMID, and I actually recommended it for a prize. I'm not entirely sure it was, my suggestions were taken forward, but I just think it's a fantastic study. And what they did was to basically take, in this particular case, metallo-beta-lactamase in Vim. They had sensitive, intermediate, and resistant, and these are the MICs to imipenem and the MICs to meropenem. They simulated human dosing. And you can see here that clearly, when it's sensitive, you've got almost total eradication of the organism. Even with the intermediate here, you can see that you have compromisation of eradication. With this next level, when the MIC is 4, you can see here that you almost get no clearance. What you have is static. And here, with the resistance, after a while, you get regrowth. And so even when the MICs are low, in an in vivo model, you still get either maintenance of the organism, but you don't get eradication. And there's another study that was done a few years earlier by the group in Paris, the authors Belay, where they took a Pseudomonas with Vim2 in, and they tried to, again, do a similar experiment. And again, you did not get eradication even with aztreonam. So this kind of shows you the antibiograms of this. And again, I would acknowledge Patrice Nordman for this. And here you have OXA48. This is what they call the Hodge test, KPC2, and NDM. And what you're looking for here is the sort of the classic, what we call clover leaf distortion of the zone. And with OXA48 and KPC2, you do get that. But interestingly enough, with NDM1, you seem to have a flattening of the zone. Now, NDM is actually not a very good enzyme. In fact, it's a rubbish carbapenemase. And it needs a lot of help to actually mediate resistance. It has help, it has a chaperone, but sometimes that chaperone is missing, and when it's missing, the MICs go right down, and this is probably why we're seeing the fact that tests like the Hodge test should not be used. And again, I acknowledge Patrice for this, and you can see here that the Hodge test was pretty successful in their laboratory in picking up these other carbapenemases, but with NDM1, it was simply not sensitive enough. Some time ago, I worked with a company called Biodisc. And Biodisc, about 2.5 years ago, were taken over by Biomuria. And whilst that was happening, I tried to get together, or to try and invent, if you like, another method with some of my Swedish colleagues at the time to try and detect metallobeta-lactamases in particular in Enterobacteriaceae, because the current E-test strip was simply not sensitive enough to do that, and also it gave false positives. And I'm pleased to say that we've done that. The paper is in press, and here we can see meropenem at this end of the strip, and meropenem plus the inhibitor, where you have an increase in the zone. Interestingly enough, we used meropenem as the substrate, which I originally didn't think would work, but it in fact works very well. Whatever system you can use, and you can use all sorts of molecular biology to try and test or detect these antibiotic-resistant genes, these carbapenemase genes. Checkpoints is very good for kind of screening normal flora, but at the moment it's still quite expensive. And you can actually use spectrophotometric assays, you can use molecular biology, but before, of course, you go down to that, you're going to have to choose a screening plate. And if you're going to either screen the patient when they come in, or indeed just have a general screening retrospective survey in your hospital or in the community. And for that, I would personally recommend that you actually use a McConkey plate with very small levels of meropenemine, something like about 0.25 or 0.5, and then you can then go on and use the molecular methods. My colleagues would disagree with me. They would use a third-generation cephalosporin. But if you think about the fact that if you're screening a population where 80% of the people actually carry ESBLs and CMYs, then you can imagine that actually you're going to be fishing for a mackerel in the sea, in the ocean. And so I think that you're eventually going to have to move into a carbapenem to make your selection process more valid. Again, I would acknowledge Patrice for this. And some countries around the world now are screening patients when they come into hospitals, specifically for NDM1, but this could also be extended to OXA48 and the KPC group of enzymes. And in France, and in China, and in Korea, and in Taiwan, and we're starting to do this in the UK now, that there is a screening process for people who have been abroad, and particularly in some parts of the world, to see whether in fact they are carriers of this. It's very important. We had an outbreak of NDM1 in Acinetobacter baumannii in Swansea, and it was well taken care of, but there was no screening system in place to start with. I'd just like to finish by acknowledging people who have helped me in my research over the years, of which I've hopefully given you some experience of. The group in Cardiff. I'm actually sort of in pseudo-sabbatical between Cardiff and Brisbane, University of Queensland. Jan Bell for her work historically on AIM 1, which I haven't spoken about, and also I'm sure you know them, the group in Paris. And so I'd just like to thank you very much for your attention.
We have time for one question, so if anyone has a burning question, please.
Can you comment on the utility of nitrofurantoin and or fosfomycin, for example, urinary tract infections with some of these pan-resistant bacteria?
It's an extremely good question, because those are two drugs that have been forgotten about. I can't comment on the KPC situation, and I'll let Yehuda talk about that, but with the NDM ones, the majority of them are sensitive to nitrofurantoin, but about 60 to 70% of the ones that we've tested are resistant to fosfomycin. We're looking at kind of fosfomycin derivatives at the moment to see whether we can actually manipulate those to make them more active, and there is some evidence for that. But nitrofurantoin is the one that's kind of been forgotten. It's quite interesting that actually it's the old drugs from the 1950s are the ones that we're relying on in 2011.